Now, our final video in the series on action potential, we're going to look at the role of myelin and how it helps to speed up the transmission of action potential compared to in unmyelinated neurons that have no Schwann cells on the outside of their axon. When myelin was introduced, we remember that myelin is simply the cell membrane of a Schwann cell wrapped over and over and over again around the axon of a neuron, and therefore myelin is almost entirely composed of phospholipid because, again, it is a cell membrane wrapped around not just once, but multiple times around the cell membrane of the axon. Now, because sodium would have to cross multiple layers of cell membrane if it wanted to get into the axon through a myelin sheath, obviously it can't cross a myelin sheath, whether it be assisted or unassisted, because again, that would be the equivalent of passing through not just a single phospholipid bilayer, but multiple layers as well. For this reason, because of the inability of ions to pass through these layers on a myelin sheath, we can say that myelin functions primarily as an insulator against electrical charge. So because of the fact that phospholipids, other than the phosphate head obviously, are predominantly nonpolar, this makes myelin sheaths very, very poor conductors of electricity, and because of the fact that the overwhelming majority of a myelin sheath is nonpolar, that means that there's nothing to attract ions to staying in one specific space around a myelin sheath. This means that in order for sodium to enter the axon of a myelinated neuron, the sodium would need to enter the spaces that are found in between individual myelin sheaths, which are what we call the nodes of Ranvier, and this has a very interesting effect on sodium, because if we consider all of the sodium that is located on the outside of an axon, we have to take all of that sodium and have it enter a much smaller surface area of the cell. Now, as a consequence, if we're taking all of the sodium that exists on the outside of the myelinated axon and moving it all into the nodes of Ranvier, that means we're taking the concentration of sodium on the inside of these nodes of Ranvier and increasing it, because once again, as long as the sodium wants to enter the axon, that means that more sodium would have to enter a very small volume of space, and if we compress the same amount of sodium that exists on the entirety of the outside of the axon into these small spaces here, the overall result is a drastic increase in the concentration of sodium in these areas. And the higher this concentration is, that means that the diffusion into the axon when the actual process of depolarization occurs, because there is an even higher concentration in these nodes of Ranvier, the diffusion inside of the axon is going to happen faster, and therefore action potential starts, that being depolarization occurs faster in myelinated neurons. And if we actually look at this process happening between two nodes of Ranvier, because there is a higher concentration of sodium inside the nodes of Ranvier, more sodium is going to enter the axon through the node of Ranvier like this, and because it reaches its critical concentration higher in order for the voltage-gated channels to open up, that means more sodium enters even faster. But because the myelinated portion is almost entirely nonpolar, this sodium is not going to want to remain in one place. So because of the faster diffusion inside, the sodium skips down to the next node of Ranvier, where depolarization happens faster once again because of the higher concentration of sodium here. So sodium diffuses in much faster as well, which results in it diffusing faster to the next node of Ranvier, and so on and so on and so on. And this is why action potential is said to skip between each of the nodes of Ranvier, which is what we call a saltatory transmission versus a continuous rate of transmission that exists within a non-myelinated neuron. And again, this is why the transmission not only moves faster, but moves in a discontinuous motion of quick skips rather than a slow and continuous transmission from the cell body to the end of the axon terminus without myelin. This concludes our series of videos on explaining how action potential works. In the next series of videos, we're going to look at what happens to action potential when it reaches the axon terminus and how that action potential is transmitted from one neuron to the next neuron across a synapse.